much, and thanks for the nice introduction. So I am a neurologist, and I do have a neurogenetics clinic, and that's what we do out in Yakima and Spokane. But the group of investigators with whom I work, this huge collaboration, so we, between all of us, have many grants, are really focused on immune mechanisms in neurodegenerative disease and trying to understand the molecular drivers of disease. So the disease that we'll be talking about is basically Alzheimer's disease, and this is a drawing from Dr. Alzheimer from 100 years ago, and you can see so these are sort of the classic features of Alzheimer's disease pathology. There is this abnormal collection of protein that's external, extracellular from the neurons, and then within the neurons there's a collection of abnormal protein, which eventually we found out was hyperphosphorylated tau. So that was over 100 years ago, and since then we've been working with this model of dividing Alzheimer's disease as really having these two pathologies. And here's a more recent image of an amyloid plaque and neurofibrillary tangles. People can have different clinical presentations, but really we don't make the diagnosis until there's evidence of amyloid. So that interaction has really driven the field in terms of finding therapeutics. And so a number of important things happened in the 80s and 90s. In the 80s, we were able to take that plaque and analyze the peptides and proteins and identify that plaque as being made up of amyloid beta, which is a cleavage product of the APP protein. So that was just on a biochemical level. And then by the 90s, we started to learn about genetic drivers of Alzheimer's disease. And so people have been following these autosomal dominant families for decades. And then finally, with lineage analysis, we're able to determine that these families have mutations in either presenilin 1, presenilin 2, or APP. And having a mutation, or I should say variant now, having a variant in these genes is 100% penetrant. So we know that an abnormal function of any of these genes is sufficient to cause Alzheimer's disease. What was more amazing was that these three genes, or proteins of these genes, are related. So here is an image of what the presenilin and presenilin 2 complex looks like. It sits in the membrane. It has 3 or 4 other protein partners. And what it does, this gamma secretase complex, is cleave substrates that are type 1 membrane proteins. So what was amazing is that the APP protein that releases A beta is cleaved by this presenilin 1 or presenilin 2 gamma secretase. And so this relationship of all these genes leading to amyloid beta suggested that really we need to focus on amyloid beta. That removing that must be the key to solving Alzheimer's disease. And so this is just an image. So this is what these proteins are. They're sitting in the membrane. They get cleaved sequentially by 2 different complexes. When they get cleaved by the gamma secretase complex is when it gets released. A beta gets released and deposits in the brain. So the amyloid cascade hypothesis largely drove the development of medications and therapies, and I should say not just therapies, but actually formed the basis for animal models. That was the readout that everyone was using is how good these models actually show amyloid. So the idea being that amyloid is created, is deposited either because of gene mutations or some other function. The amyloid beta is directly toxic to neurons. It causes them to make parahelical filaments and then causes neural dysfunction and death. And the idea being that presenilin mutations were a toxic gain of function. They produce too much amyloid beta. So, like I said, models and trials were based upon the amyloid hypothesis. If you can remove amyloid, you might do better. The mouse models, flies, worms, are all based on expressing the presenilin and APP gene mutations. What's interesting is that these mouse models, just expressing one of these genes is not sufficient to cause Alzheimer's disease in the mouse. These are crazy mice that have at least 3 disease-associated mutations within the mouse in order to produce the pathology that we eventually see. So there's a little bit of challenge in using these mice going forward. Not going forward, but in general. And we developed therapies. And they have not yet been successful. I know there's some excitement about one of the drugs, and we'll see what happens with that, but generally they have not been successful. So a number of factors about drug therapy have been considered, one of them being we just need to start earlier, and so there's a lot of work going on with biomarkers to try and identify people early, maybe even preclinically, so that you can target anti-amyloid therapy. But another really interesting development in Alzheimer's disease, really in the last 10, 15 years, has been the appreciation that not only does presenilin-1, presenilin-2, and APP genes cause Alzheimer's disease, there are other genetic risk factors. So here is a graph, and so on the y-axis is the risk of Alzheimer's disease. So high is here, fully penetrant, low would be extremely low risk. And then on the x-axis is the frequency of the variants. So you can see extremely rare are these autosomal dominant variants, and then out here are these common variants that 30% of us might have, 50% of us might have. So when we started doing GWAS studies, comparing tens of thousands of folks and looking for risk alleles, that's when we were able to identify some of these very frequent but extremely low risk variants. So this is like on the order of odds ratios of 1.1, 1.2. So the idea is that having just one of these is not sufficient, but it's probably a collection of a number of these. People have their own profiles of risk, and it'll include some aggregate of all this. What was very telling then is that having these gene variants and I should be very clear actually, these are not gene variants. Out here are gene variants, so variants within the express coding or a regulatory region of the gene. These are actually just SNPs. So these are single nucleotide polymorphisms that are within a region that includes these genes. So we're actually just sort of guessing that the genes are associated with the SNP, but so let me just make that one caveat. Okay, so the genes that we associate with the SNPs, though, fall into a number of cellular pathways. So not only production of amyloid, but also regulation of immune function, endosome function, cholesterol. So that really sort of broadened our idea about what are the pathological mechanisms on Alzheimer's disease, and are there pathways that we can target beyond amyloid beta? So I'm just going to tell you what the take-home things of what we've learned recently, or at least over the last 10 years, is that immune pathways are relevant to both familial and sporadic AD, that disease-associated genetic variants can actually act in cell type specific ways. Isolating cell types from Alzheimer's disease autopsy archived brain can show numerous cellular subpopulations, which is important in understanding biology. And then computational approaches can be used to sort of take that data that's taken from end-stage disease and actually transform it into something that tells us about the dynamics of disease progression.
Can I ask a question?
Yeah.
What's the prevalence of familial Alzheimer's?
It's very low. So it's maybe less than 1% of all Alzheimer's disease.
Good.
Yeah.
It's good to hear
Yes.
that my father has Alzheimer's.
Right.
Macro.
Yeah, so these autosomal dominants are extremely rare, and then there's a whole slew of people that have a family history, and if you have a family history of someone with Alzheimer's disease, you have a slightly increased risk.
Okay, good, thank you.
Yeah.
On that note, those variants that you showed there, are those all germline variants? Or is any of
Yes.
this somatic?
No, well, okay, so that's actually an extremely important question. So these here are likely germline. They're taken from blood, so these are all variants found in the circulating system. These are not variants found when looking at tissue and doing genome analysis of tissue. There is evidence, though, of somatic variation in the brain. People have been looking at whether or not there's changes in APP, even presenilins, and so the whole idea of even disease starting through some somatic change in tissue is an important one, but not what we're discussing.
So Dr. Elmo means safe.
I don't know if I'm safe, but there's hope.
Maybe this would be where this becomes relevant to neurosurgery, so you just take out that somatic. I was trying to figure out where I was going to bring in the nurse.
This is fascinating. It lists 60% of people developing dementia by the time they're 80.
This is so these are the things I'm hoping you'll go away with, and if not, come back to this slide. Okay, so like I said, we learned a lot about Alzheimer's disease from the familial cases, and there is this great history here at the UW of the presenilin-2 gene mutation. Dr. Tom Bird had followed these families for decades, and then finally they identified the gene presenilin-2. And here's an example of a pedigree of these families. This is just incredible. So, we were able to, or Tom was able to identify at least 4 or 5 generations here, and everyone in black has the disease, Everyone is clear, does not. And you can see that, and a number of things you can see just from the pedigree alone, that there is male-to-male transmission. So you know it's not an X-linked disease. And it seems to be autosomal dominant. Here's an image of what the presenilin 2 gene looks like. And so these are the transmembrane domains. Here is where it is catalytically active. What is interesting when we look at where gene variants are associated with disease, where they occur in the gene, we're looking at functional domains. Is there a hot spot? In the presenilin 2 gene, and to some degree presenilin 1, the mutations are sort of all over, which makes it difficult to really focus on one particular region. Okay, so here is the cartoon of what we have always considered to be important about this relationship. APP is here, it gets cleaved by the presenilin gamma secretase, it releases A beta. But there is a whole host of things that this cleavage also does. Cleavage of this protein here releases an intracellular domain, which is very important or drives transcription. So this is extremely important in regulating notch activity, and notch is really important for development. And cadherin, there's a number of signaling activities that happen through the intracellular domain. So is that important? Well, notch knockouts are lethal, and these proteins also to some degree regulate inflammation. I'll talk about that in a second. So does this matter? Well, so here is a graph of the activity essentially of patient cells, fibroblasts, that either have mutation in presenilin 1 or presenilin 2. So let's start with the presenilin 1. Basically, the dark blue is the control. And what you see is that the amount of this intracellular domain being produced is decreased in many of these compared to the control. And in the presenilin 2 cases, all these colors here, these are the mutations, also have markedly decreased intracellular domain. And we've actually shown that the activity of notch is hampered in one of the presenilin 2 mutations. So we know that there is a functional consequence on other activities besides A beta from familial Alzheimer's disease gene mutations. So this is what I was talking about. What was really striking when we were starting to look at the biological activities of these presenilins is not only do they cleave a number of substrates that are important also for neurons, neurite outgrowth, axon survival, but also regulation of the immune system. So genes, proteins from called TREM2, CSF1R, interleukins, interferon receptors, these are all substrates of the gamma secretase complex. So where is the evidence in humans that the immunity is really a big deal? This is a PET image, so this is a labeling activated microglia supposedly, but it's, which is sort of a generic term. But it's, so you can see here in the normal brain, there is, some low degree of activity, but in Alzheimer's disease brain, there is a lot of signal from this PET that shows that microglia are active in the Alzheimer's disease brain. So that was sort of one, real-time clue. And then the genetic evidence, we already talked about genomic studies that have risk loci that include innate immune molecules. But also in the last 10 years, we found that variants in TREM2, which is a microglia and macrophage gene, variants in that gene alone can increase your Alzheimer's disease risk by 3 to 4-fold. So having an abnormality in the immune function is enough to drive increased Alzheimer's disease risk, which there's more evidence that this is important. So how could that happen? Well, there's a number of ways that we can imagine the innate immune cells in the brain impacting neurons, and this is just a very simplistic cartoon. Basically, the microglia and the neurons, so the microglia are the innate immune cells of the brain. They're monocytes, basically, resident in the brain. So microglia and neurons are constantly interacting. Microglia are always sort of remodeling the synapses. That remodeling is extremely important during development, but occurs even at the, sorry, it's been a big mess.
No, don't worry about the mess. How about you?
Yeah, so thanks. So in a healthy state, microglia and neurons are interacting, and then in a disease state, they can release cytotoxic molecules that drive neuronal death or neuronal injury. They can start abnormally pruning the synapses, and there's evidence, again, from human tissue that suggests that. And then they may have impaired ability to clear debris. So many different behaviors by these innate immune cells can impact neural health. So we developed sort of this working model of presenilins that says, yes, the amyloid beta is probably problematic, and yes, amyloid beta is a product of the presenilin genes, but really the picture of Alzheimer's disease mechanisms really has to be put into a larger context. And so in addition to amyloid beta production, we were hypothesizing that impaired immune pathways are also important for driving the disease. This becomes a bidirectional communication between A beta and immune function, and that mutations in presenilin not only cause amyloid beta, but also impair the immune function. So what we did a while ago is to create a presenil-2. Well, we actually used the presenil-2 knockout mouse, isolated microglia from this knockout mouse, and just assayed the behavior of these microglia in a dish. And what we showed is that the presenil-2 knockout microglia, compared to the wild type, release higher levels of inflammatory cytokines, of which IL-6 is an example, when you expose it to a canonical TLR4 molecule, LPS. We found that in addition to releasing higher levels of cytokines, these microglia also have increased NF-κB activity, again another canonical driver of the inflammatory state. So at that point we were trying to figure out how could presenilin be modulating this, and so we started to look at what were being identified recently as really quick modulators of the immune system, that being microRNAs. And so we had looked at one of them, well, we looked at a number of them, but one of them is microRNA 146, which inhibits the expression of the proteins that are responsible for driving NF-κB activity. And consistent with there being increased NF-κB activity, the presenil-2 knockouts also had decreased levels of this inhibitory microRNA. So that was developing as a possible way in which presenilin could independently drive inflammation. So then we created this transgenic mouse, we as in Bryce-Over. Basically, it's a human presenilin-2 transcript that is flocked with a stop sequence. And so once the animal is crossed to a basically the animal expresses the human mutation. And what we did was put that human mutation on a heterozygous mouse, so that instead of having multiple alleles of presenilin-2 and then trying to figure out what's going on, we tried to create a situation that really closely mimicked the familial AD people. So this is just a graph that tells you that the protein is expressed and you see what you expect. So the first thing we did was try and figure out if this human mutation impairs gamma secretase activity, in which we showed that in fact in the microglia they do, that there is decreased gamma secretase activity, and that was important because we attributed some of the immune dysfunction to abnormal gamma secretase activity. We have now shown that these microglia also have increased NF-κB activity, increased expression of these cytokines, and again this is from in vitro microglia. And interestingly, they also were more aggressive about internalizing amyloid beta. So this is this is a picture of the facts, and this is really a measurement of how much A beta is internalized in the N141 in the mutant microglia compared to the control. Okay, so now we've only really been talking about cells in a dish and how relevant is that. And so what we did, we tried to figure out what is the impact of this mutation on the global inflammatory state, or at least systemic inflammation and the response by the mice when they are exposed to an inflammatory stimulus. So first we looked at the baseline level of some of these genes and found that even in the absence of any stimulation, the mice that carry the human mutant allele have increased levels of these pro-inflammatory cytokines, as well as this molecule TREM2, which is a critical sort of driver of the inflammatory state of microglia. So even at baseline, they are sort of abnormal, and this would be the similar state of what humans having this gene mutation would be. And then when we expose them to LPS with intraperitoneal injection, which really ramps up the immune state, we again see that there's really a more exaggerated release of some of these cytokines in a mutation compared to the control. So this is what we are sort of working with now when we think about familial AD, that the familial AD mechanisms are probably not just amyloid beta, they probably do also reflect some dysfunction of the immune system, which is going to be important when you try to remove amyloid beta. In these cases, are you really solving all of their problems? Or is there some other dysfunction that we have to address? Okay, so these were cases here, these high risk, low frequency. And then if we kind of go down here, these are the rare variants. So these are actually coding variants, so in the gene itself. And so we started working with Jessica Young on one of these genes here, SORL1. So she came to speak to you, I think, in spring. Okay, good. And she had probably talked to you about the work that they've been doing in vitro using iPSC neurons. And so just to remind you, SORL1 is a intracellular trafficking protein. So it takes things from the Golgi, from the endosome. An abnormal function of SORLA increases the release of amyloid beta in some of her neuron models. Now, because SORLA actually manages intracellular movement of A beta, we also hypothesized that that same thing was happening in microglia. So what we needed to do at that point was create microglia from induced pluripotent stem cells. We can compare them with Jessica's neurons and then perform assays on the microglia derived from human patients compared to the neurons, and then co-culturing them to see how they interact. This is from Jessica's data. What she first showed you was that loss of SORL1 causes less efficient internalization and trafficking of amyloid beta. So this is a knockout line of iPS-derived neurons, a knockout SORL1 line. And what we see is that the amount of A beta internalization is impaired in the cells that are absent SORL1. And then conversely, if you overexpress SORL1, you can drive increased transit of amyloid beta into the lysosome. So, we first had to figure out how to make microglia from the iPS, and so we actually developed, we had one protocol that we developed. It was not as robust as some of the protocols that have been coming out more recently, but this is what we now do. We had a postdoc that went to the lab of someone at UCI who had sort of perfected this protocol, and basically you start with induced pluripotent stem cells. You then dry them to a hematopoietic precursor. Here's an image of what they look like, if anyone's interested. This is about 12 days after driving these cells to hematopoietic precursor. And then from here, we add a cocktail of molecules that we know exist in the early CNS. So this is TGF-beta, CSF, IL-34. These are molecules that we know are critical for microglia maturation and development. And then so after about a month or so, we have these very beautiful microglia that are now, expressing the human genome, not the mouse genome, which was really important. Just to validate that we have the right cells, what we can do is look for microglia-specific genes. So here is a graph of gene expression from monocytes, two different differentiations that did not work, and then the differentiation that produced microglia, and you can see that it has really elevated levels of this gene, hex B, which is a microglia-specific gene. And then other genes such as TMEM119, Q2RY12 are particularly enriched in these microglia compared to monocytes. And to some degree, microglia share many features with peripheral monocytes, macrophage, but because they are CNS resident and they also don't turn over as quickly, it is important to be able to find a protocol that creates microglia, real specific microglia. And then we can just demonstrate that these cells don't just look like microglia based on gene expression, they also have functions that are critical that we're going to be using as readouts, such as release of inflammatory cytokines and then phagocytosis of amyloid beta. Okay, so when we then expose the microglia that were then derived from sorrel knockout mouse, in contrast to neurons, these microglia don't have any impairment in their trafficking of amyloid beta. And that was a surprise, but there are a couple things that really showed us that SORL1 has cell type specific effects. So if we are going to target medications towards promoting SORL1 behavior, what we really need to do is make sure it's working well in the neurons. And so, when does that become an issue? Well, if you're using sort of neurotropic AAV to do genetic treatments, knowing the cell type is important. Okay, but the issue is the 60% of us that are going to get Alzheimer's disease, all the Alzheimer's we have, and mostly in the population, are people that have this type of risk, the risk that we all have, but an aggregate of this risk. So how do we study this? So the genes that we've talked about just now are variants that occur within the coding region of the gene that have a measurable change to the protein. So you can guess what the function of this protein is. You can assay that function and really understand how does a variant cause disease, or at least cause problems with the protein. The problem is most of these variants are occurring in the region of the genome that are non-coding, and often not even, like I said, they're not even within a regulatory region of a particular gene. Very few are. So how do you then assess the impact of these variants when you don't even know what to actually read? And so this requires alternative approaches, and what it requires also is starting to amass data from thousands and thousands of people. You know, we can't use a mouse model to model this. We really can't use patient cells unless, again, you're going to be using thousands of cells. So what we've been able to do is, or what we've been trying to do, is try and use some of these omics approaches to study complex disease. So we all have some huge aggregate of genetic risk, and so we know that there's this profile of genetic risk of people that have AD and people that don't. But then how do we go from here to here? That's our problem. So what we can do is measure these quantitative traits, these sort of proxies for disease progression, the biology of disease, and these quantitative traits, which are the omics, are gene expression, protein and metabolite levels, metabolomics, the actual cell function, if we were to introduce some of these into a cell, and then epigenomics. So how are the chromatin structure, the methylation of the genome, how is that impacting cellular pathways? And so do these variants actually change the way the chromatin is sitting together, the chromatin is opening up for transcription? So some of the questions that we can start to look at using this broader approach is that we can identify what is the normal brain gene signature compared to the Alzheimer's disease gene signature. So you can see what genes are abnormal here compared to this cohort, and then start to understand biology. So we start with brains. This is a different study, but you start with archived frozen brain, and then you can measure RNA from these brains, and then start to look at what are the differentially regulated genes. And so this is just an image. You know, in prefrontal cortex, there's a large amount of differentially regulated genes in Alzheimer's compared to control. And notably, this type of analysis, really requires thousands of patients. And so looking at the actual gene changes, we found that the pathways that we suspected because of risk alleles seem to be truly abnormal. So immune, cholesterol, endocytosis, all of these pathways are transcriptomically different in the Alzheimer's disease brain compared to control. And that is information we have when we take a chunk of brain and then we do the transcriptomics. The problem is which cell types are actually responsible for these changes. Again, we have some clues that cell type matters, but if we're going to actually try and model the disease and target, we need to know, what exactly is happening. And so, you can take this little mess of brain tissue and then sort it out, or not sort it out, but actually separate it out. And so what we do is take frozen brain. We then prepare single cell or single nuclei preparations, and then using the 10x system that allows you to barcode each cell, you can barcode each cell and then measure the transcripts of each barcode, and then get a sense of what the transcriptomics are for each cell type. So here's an example of what the readout looks like when you just take a piece of brain and you analyze the using the single cell approach. What you see is that each of these little dots is a cell. That the cells sort of automatically self-organize into different clusters, and so they're clustering based upon their transcription profile. They cluster, and then if you look at the gene signature within these groups, you can identify the cell type. And so these are oligodendrocytes, neurons, microglia, and one first thing that you can do is try and see how do the dynamics change between a control and an Alzheimer's disease brain. So, you can create a pie chart, essentially of the different cell types here and say, well, there are this many inhibitory neurons, this many excitatory neurons, this many disease-associated microglia, that's called DAN microglia, and then, different types of glia. And then you can look at the Alzheimer's disease brain. Which kind of neurons are we losing? Are we losing the inhibitory neurons? Do you see this explosion of abnormal microglia? And then you can, focus more of your study on these particular subcell types of, or subclusters of cell types.
How are you defining disease-associated microglia from the transcriptional signature?
Yeah, so this is actually, this is a phrase that's been given a couple of years ago using mouse models. And so, actually using single-cell approaches in an AD mouse model, you can identify which transcriptional programs are active.
But Suman, wouldn't the specific tissue region that you're preparing
Yes.
for matter?
Yes, it absolutely matters, because what that study showed is that these microglia are actually more active when they're around plaque, the disease associated. So it does matter, and this is one of the big one of the big challenges of using this approach right now in human tissue is that you don't have good spatial resolution. That is changing. There are now methods to do spatial transcriptomics, which is crazy. So you can actually take this tissue, put it on a slide, and really do sort of the same sort of thing where you can get gene expression regionally. Very expensive, still being developed. Okay, so here's an example of how far we've gotten in the field using single-cell transcriptomics. So this is a paper that came out just a couple of months ago, and this was sort of our version of a tour de force. Out of using only 48 individuals, they were able to do single-cell analysis, and really interestingly, that 48 individuals only results in about 80,000 cells. So we are distilling a lot of information from very few cells. And so, what they were able to look at the most highly expressed genes in different cell types, microglia, excitatory, inhibitory neurons. But there is some interesting information you can glean from this. So here is a map of gene expression, and then on the x-axis, these are different types of cells, excitatory, inhibitory, microglia, et cetera. What you see is that when you compare the early disease brain to no pathology, there are pretty obviously cell type specific changes in certain genes, changes that are not shared with other cell types. But if you start to look at late versus early, all these cell types upregulate similar patterns of genes. And so looking at the ones that are shared, it's what you'd expect at sort of the end stage of disease, protein folding, autophagy, et cetera. So, things that we sort of suspected, you can actually see bear out here. But the fact that all these cell types eventually show this is sort of interesting. Looking at this data and trying to, focus, going back to the innate immune cells, the microglia, one unfortunate thing with single cell is that, and also in the brains, you get very few microglia. So out of this cohort of 48, there's only about 1,000 microglia. So trying to get information from 1,000 microglia is a little challenging. And so, while we've been doing this type of analysis, we knew we really had to focus on trying to enhance our collection of microglia. And why is that important? Because in that 1,000 microglia is this really diverse group of phenotypes. And so microglia are antigen-presenting cells. They actually interact with oligodendrocytes. They can modulate exosome movement. They release inflammatory cytokines. And so, when we're going to target the immune system for therapies, we have to know what exactly we're targeting. And there's, interest in trying to use immune modulators for Alzheimer's disease, for Huntington's, and that's great. But really, that's such a large hammer. When really what we want to know is there not enough of the good behavior, or is there too much of the bad behavior, or is it a balance, and which cell type is really doing this? And so in order to understand that, you need to be able to interpret the transcriptomics from that particular type of microglia. So what we did was we, again, made these single nuclei preparations, but then tried to use a FACS sorter to really enhance our amount of microglia. So we used this nuclei marker PU.1 and sorted the nuclei. And this is just a picture color you can see. So the guys in yellow are the neurons identified by mu n, and the guys in green are the microglia. So sorting these, and as you can imagine, this sort of process was quite involved, because one of the big challenges also with this approach is that when we're using archived tissue, the tissue itself may not be the quality that you want. So we did a lot of this work first in the mouse, and that was great. It's easy because you sack the mouse, and then you can take the brain, you can even sack the mouse and freeze the brain to see if you can figure out how do you make these guys thaw better. But really, when you're dealing with human cells, it's a lot more challenging. Okay, so did it make a difference? Well, in our hands it has. So now if you just take that cluster of cells that are only PU.1 positive, so you really focus your sequencing on these cells, you can see that even within that microglia group, you're starting to see heterogeneity, so different clusters of microglia. And when we do gene expression of these groups, and so these groups here, 1, 2, 3, 4, so they are ordered really by their highest differentially expressed genes. And so what we were able to do is try and get some idea about who these guys are, and this microglia 1 gene group is likely more of the inflammatory type compared to the others. And so this is the type of information we want to know. When we look at Alzheimer's disease and we say the microglia are abnormal, what pathways are specifically abnormal? Okay, but this is all end-stage disease. So how much can we really learn from this? And this is where our friends from SAGE become very important. So what Samit Mukherjee and Ben Logsdon have been doing, and a lot of other people have been doing, is trying to look at the transcriptomics and see how they order along an axis, with the assumption being that there are different degrees of disease even within the same person. So this is sort of a cartoon of how you do it. So you start with people with Alzheimer's disease, control, and some other random disease as a disease control. You do the RNA analysis, and then using this process called manifold learning, which is also this, if anyone knows what the monocle program is very similar. And so then you can use this computer and this computational approach to order the transcriptomes of each of these people along axes and start to see where they cluster, start to see where they branch. And you can use this information to say, what are the transcriptional changes that occur at the branching point when all of a sudden you go from normal to Alzheimer's disease, or normal to another group of diseases. So that's sort of my simplistic version of this. And so by mapping them all on these compressed axes, you can start to see how these people relate to each other transcriptionally. Okay, so can we test that? So this is work that they did, Ben and Sumi did a while ago. And so what they did, actually I'm going to go back here. So this is sort of a difficult image to look at because they've used very subtle changes. It's like Martha Stewart colors, but so the each of these dots is a person, and they are labeled based upon what their Brock stage had been. So this is all archived tissue. So an individual was diagnosed with Alzheimer's disease or not, and then given a Brock stage. And so remember, Brock stage, well maybe not, Brock stage is the amount of Alzheimer's disease pathology, basically. 6 is the highest, 0 is the lowest. And so what they did was look at these transcriptomes and again allow them to be ordered and use manifold learning to see how they relate to each other. And what this is supposed to show is that the people that have a high Brock stage tend to cluster over here. The people that have a very low Brock stage tend to cluster on this side. And this is this axis is sort of disease progression. So the way that the manifold learning is interpreting the progression from this group to this group is consistent with the progression that we have decided based on pathology. So on the y-axis here is the inferred stage of disease based upon just computational analysis of the transcriptome. And here on the x-axis is the Brock stage. And so there's a nice relationship there that suggests that we can sort of understand something about disease progression just based on the transcriptomics alone. So, right. We can study, so this allows us to look at dynamic processes. So that was in bulk tissue. Now and this is tissue that is from the AMP-ED project, which is this big consortium of different universities that have their own banks of tissue, Alzheimer's and non-Alzheimer's disease, right? And so Ben was interested then in trying to further delineate what are the changes you can see in cell types, and so they deconvolved that bulk tissue. So they computationally decided what were the changes that happen in neurons and microglia, and there are some other interesting points that they were able to see. So here the cells that come from ED patients are in red, the cells that come from control people are in green, and what you can see, maybe not so great, is that again there is a sort of progression of neurons that come from control people compared to neurons that come from AD people. That makes sense. The microglia were even more interesting, of course this is my bias, but that not only is there a progression of disease as you go from control to Alzheimer's disease, but that there are different clusters even within the AD group. So, what is the Alzheimer's disease occurring here versus the Alzheimer's disease occurring here? And are these meaningful differences? Is this something we have to pay attention to? What we're going to then do is try and look at what are the gene changes here. So that's using deconvolved data from bulk. And so now what you can do is use the same approach of pseudotemporal ordering on the single cell data and really get finer resolution. So this is work that is a combination of some of the single cell microglia data that we have and the data that Ben was able to extract from that big single cell paper I showed you, focusing just on the microglia. So in this group, they had very few microglia, and so what they were able to see that their microglia clustered basically into a few clusters. The microglia 0 cluster, so these are all the cells within that cluster, really are sort of the more benign. They don't have anything terribly abnormal. If you plot the female microglia and the male microglia, they're all just sort of one big ball. There's not much of a difference. The microglia 1 cluster was the one that they called the disease-activated phenotype, and there is some controversy about how disease-activated they are, but for the sake of just showing the value of this, we'll go with it. So these transcriptomes, suggest that they are different and that they're disease, that they may be affected by disease. And what's amazing is that this shows you that there is a sex difference between the activated transcriptomes of men and the activated transcriptomes of men versus women. This is work that's been, this is a concept that's been appreciated more and more over the last 10 years or so. In vitro studies of microglia, rodent models of other immune cells show a sex difference, which is why now everyone has to double their rodent colonies because everyone has to do men and female, males and females. Okay, so that was information telling us that there are differences between men and women. When Ben then looked at just the microglia, the disease microglia, just in females, he can see that in this group, there really is, again, a progression of control to late AD. So the conclusion from that is, in women, the disease microglia are changing, and you can learn something about the disease progression. In males, we don't have that same association. Okay, so that's great, but what can we do to actually then understand what the impact of this is using our models? Like I was saying, we're not going to make 1,000 iPSC models. So what we've tried to do is at least try and bin some of this. So, these GWAS hits are all over, and like I showed you, these all represent different pathways, and so you could hypothesize that people that have an aggregate higher amount of endosome abnormality may have more of an endosomal-driven Alzheimer's disease, or people that have more of an immune genetic risk may have immune pathways that are abnormal in AD. And so what we can try and do is at least pick the people that have a particular type of gene risk so that you can get your N smaller and you can really have hypotheses to test and not just shotgun everything. So what we did was we gave these cases of polygenic risk score. And so again, this is not stuff that we're using in the clinic. This is taking cerebellum, genotyping across the SNPs that we know are disease associated, and then basically doing a weighted analysis to figure out, how much risk does someone have. What we then did was focus on just the SNPs that are associated with the endosome function. And so they get a endosomal PRS. And so doing that, you can see there's a spread. So these are controls in the black, and then these are sporadic AD. And you can see that. So I think I took it out. Basically, there is a higher endosomal risk score in the purple cases compared to the black cases. And then what you can have then is you can have on the extreme people that have a really high endosomal risk, people that have a really low endosomal risk. And then we can say, okay, our hypothesis is that if we then take cases from these tissues compared to these tissues, we expect there to be problems with the endosome function. Because again, like I said, we are assuming these genes are related because they are in the same region as the SNP, but we don't actually know for sure. And so that's what we're doing now. And so this is this huge multidisciplinary team. So yes, I am a PI on this huge grant, but many PIs. So this is sort of how it works. There's a brain tissue. Dirk Keen in his lab then takes a piece of meninges. From there, he prepares fibroblasts, and then they reprogram the fibroblasts to become IPS. And this is work that Dirk and Jessica actually published this past year, this protocol. And the value of this is that now we've created induced pluripotent stem cells from the same person in which we can get single-cell transcriptomics, pathological data. We can actually interpret this together. And then so from here, we can make neurons and microglia and do functional assays. And at the same time, we take that cortex, we do transcriptomics, we can now also study the epigenomics, and then try to integrate this data. How does this gene transcription actually predict changes in function, et cetera? So that is our attempt to try and study sporadic disease without having to study 10,000 people at a time. Okay, yeah. So that was my son, and so he's animation by Bridge Barrington. I'm going to give him credit. I took out a lot of them earlier, so you should be glad that you didn't have seizures. So this is team science. So the folks that have been working on the presenilin and the other sort of immune in vitro stuff are here. Carol Smith has really been fabulous in the case, all these folks. Kevin Green is the one who was actually for a while coming in the middle of the night to get pieces of fresh tissue from Dirk in order to do single cell transcriptomics. And that's really beautiful dedication. I probably would have kept making him do it if we thought we could get enough tissues at the time, but if you're getting tissues real time, you're just not going to get the big enough N. So Kevin then did spend quite a bit of work trying to figure out how are you going to isolate nuclei from brain, from frozen brain. And then the omics team, so Liz Blue is a geneticist, Michael Dorsch did some of the gene sequencing, Gwen Garden is an important part of the team, and the Sage folks did all the analysis. Brad Rolfe is a genetic counselor who's been so helpful to me to sort of figure out the SNPs and putting the SNP profiles together. And then we have collaborators. And then I wanted to mention, so if there are any trainees here, probably one of the most important parts of the team are the mentors. And so without them, it's really hard to move forward. And these are the people that give you inspiration. These are the people that are your sounding boards. And these folks have been incredibly helpful and supportive. So that's it. Thank you.
Great. Questions here? Right behind.
question, sorry. One of the things that you mentioned that struck me was that the vast majority of the low-frequency SNPs are non-coding regions, right? And so, but have you guys overlaid those with, you mentioned that they were also not in regulatory elements. You've overlaid them with ENCODE data or ATAC data that show that they're actually not in sort of annotated regulatory elements. So do you guys have a sense of what that means? Are these like unannotated regulatory elements or unannotated genes, or what's the...
Yeah, exactly. So that's really important because you can still distill information if you know where they are. So the top hits that we've been using, many of them actually are in enhancer motor regions. And what Liz has actually been doing is trying to create a sort of even super SNP profile where you give extra weight to SNPs that are in the enhancer regions. And this, as you may know, that's even still pretty simplistic when we're just going to be looking at promoters and enhancers, because now we know there's all this long-range regulatory effects of SNPs. So the next phase of this is going to have to be things like doing chromatin mapping and looking at how much these SNPs are actually interfering with enhancing or promoting of genes throughout the genome. So that's an important point.
I have just a curious question, probably the stupidest question. What is the phenotype of an Alzheimer's mouse? What do they do?
Yeah, they don't lose their keys.
Yeah.
So the problem is, and this is where the sort of the readout comes. So for a long time, the readout has been, do they make A beta in the brain? And then you can make drugs to remove A beta in the brain. But they do have a behavioral phenotype, so they do have memory problems. They do have a neurodegenerative phenotype, some of them. There are different types. There is one that it's called the 5X mouse that actually has, many mutations, and those guys die less than a year. But they have memory, behavior, anxiety, motor phenotype. It's difficult to map that onto a human.
Yeah, right. It's like all mouse models.
Yes, like all mouse models. So, and I don't want to badmouth the mouse. They've been wonderful workhorses.
Yeah.
So they still have a very important role. To some degree, can you humanize them? The people at JAX have been trying to use some of these low risk alleles and somehow incorporate that information into their mouse models. And they have something where they've tried to generate genetic diversity within their mice to create better models. But
Mike Williams?
Yeah, so a couple questions that I think are related. The first is, how do you fit this into the standard clinical and research model now, which is we want to identify pre-symptomatic patients. And the second is, what lights the fire? What's the stimulus?
Yes.
Because if you have any hope at all of treating Alzheimer's disease, you've got to find the 30-year-olds in this room who are predisposed to develop it. Those of us who are full professors, it's too late.
Yeah. So that's really important. I think the reality of how we go forward in the clinic is, I think, still difficult, at least for me to imagine, because I think when we all first started this, this precision medicine, and we used to have these slides that said different colored people and you spin them up, and which work great for cancer, and it's probably not going to work as well for diabetes and Alzheimer's disease. So I think the reality of this information being used in the clinic on its own may be low. Maybe in the future what you do is you get the genetic profile, you take a piece of skin, do some functional assays, and then say you have a slightly higher risk of Alzheimer's disease. Now, whether or not you can do it for genetics, and then the problem is, so you might find out that someone has a higher risk. But if we think that there is a stimulus that starts the fire, does it matter if you have this, aggressive risk if you never have the fire to start? And then how do you treat it? Because you have to identify the fire. And we have not yet identified the fire. Now, there are people who believe that the amyloid beta peptide itself is the fire, and that may very well be true. So on a conceptual level, getting rid of the amyloid may get rid of that little irritation of that little pebble that's irritating. But what I think has been useful is saying, maybe there is this little fire, but we're not going to find it. So let's try and curb it, and at least try and stop the way it promotes degeneration. But it's true, know, there are ways you can think about this as the sort of inciting phase, and then there's the cellular phase, and so really what we're trying to do is interfere at the cellular phase.